Today, we're going to talk about some infectious agents that don't fit anywhere else, so I call them unusual infectious agents. But as you'll see, they do have a common theme among them, and that is they address this question: what's the minimum genome size needed to sustain an infectious agent? Or could you have a genome, an infectious agent, without any genome? And viroids, satellites, and prions, which we're going to talk about today, answer these questions. So first, let's talk about viroids. These are very interesting because they consist only of RNA. They don't code for any protein. They have no protective coat either. There's no capsid, just a naked piece of RNA that doesn't code for any proteins. And we call these viroids to distinguish them from viruses. And there is a database, the subviral RNA database. So these viroids are also called subviral RNAs. 1,900 different sequences to date. So this is a viroid here at the top. It's a small, circular, single-stranded RNA. And as I said, there are no protein coding regions. But when you take this viroid RNA and you put it into a plant, it replicates. And you can see these viroids tend to have extensive base pairing. So even though they're single-stranded circles, they're more rod-like in appearance because they have a lot of base pairing. There are two families. You know, we've been talking about classifying viruses into families throughout this course, like Picornaviridae and Orthomyxoviridae. The viroids are also placed in families. There are just two families, Pospiviroidae, and these replicate in the nucleus, and Avsunviroidae, they replicate in chloroplasts. So you can see DAE is at the end of the families, just as if for viruses, it's just that the first root is different. Pospiviroidae and Avsunviroidae. They're distinguished biologically. So the first viroid discovered is called potato spindle tuber viroid, discovered in 1967. And these have very interesting names, these viroids. And that's the prototype for the smallest known nucleic acid-based agents of infectious diseases. So this answers the question, what is the smallest possible genome for an infectious agent? The viroids answer this question. Potato spindle tuber viroid is 350 nucleotides long, but there are smaller ones as well. Now, some of these viroids, they all replicate in plants of one kind or another, and some of them are harmless to the plant. We don't pay any attention to them, but some of those viroids cause important diseases of crop plants, and these are crops that we grow to feed people, so they are a major concern. And you can see in this picture the effect of a viroid on a tomato plant. So on the left is a healthy tomato plant after so many days of growth inside, and the other three plants have been inoculated with 3 different viroids with different virulence. You can see one with mild virulence. It stunts the growth just a bit, intermediate and highest virulence. So the main effect of this viroid on the tomato plant is to stunt its growth. So you can see that would be a problem for tomato growers because you're going to have a small plant, fewer tomatoes, and smaller tomatoes as well. So attempts are made to try and prevent the spread of viroids. These are some of my favorite ones. As I said, the names are pretty neat. Kadang, coconut viroid, which causes a lethal disease of coconut plants. You can see the coconut tree on the right. The leaves are all yellow. And if you like pina coladas, this viroid makes you sad. Hop latent viroid. This is a hop plant. It's no symptoms typically. No bad symptoms. In this particular one, you can see some yellowing of the leaves, but for the most part, you can go ahead and make beer out of these viroid-infected hop plants. And then we have apple scar skin viroid. It causes mild symptoms, mainly in the coloration of the skin. And if you've ever seen these in a supermarket, or if you ever do, now you'll know what's causing this mottled look. And they're fine. They don't taste any different. There's nothing wrong with them. You're not going to get a viroid infection if you eat them, but people don't like to buy them so they often don't go to market. Viroids don't encode any proteins, so of course they don't make mRNA. There's no purpose in encoding an mRNA. Their reproductive cycle consists entirely of the viroid RNA and its replication. As I said, these RNAs have extensive internal base pairing. They look like rods. If you do an EM of these RNAs, even though they're circular, they're all base paired, so they look like rods. Some of them contain what we call a ribozyme. This is an enzyme made up only of RNA. We'll talk about that. That ribozyme is essential for replication. So this is a very famous enzyme that resides entirely in RNA. It's pretty cool. Now, when we talk about viruses, we like to talk about the fact that viruses are parasites of the host translation machinery. In other words, all viral genomes have to be made into mRNA, and then the mRNA has to be translated because viruses can't make proteins on their own. Viroids, in contrast, they don't make proteins, they don't encode any. They're parasites of the host transcription machinery because it's in fact the transcriptional machinery of the host that replicates the viroid genome. Remember, the transcriptional machinery consists of the enzymes and other proteins that take a DNA template and make RNA out of it. So our messenger RNA is made by a transcriptional machinery of the cell. And the viroids use that machinery in order to make their genomes. Here are representatives of these two families of viroids, the POS-P viroidae at the top, and the prototype is potato spindle tuber viroid. And you can see it's a rod-like molecule. And on this are marked different regions. And over the years, what people have done is to mutate the viroid RNA genome and put the mutated molecules into plants and ask if you change the bases in a certain region, what does it do? And you can see there is a highly conserved region in the center, which you can't change much. It ruins replication. There are variable regions which can be changed, and then you can see there's a region involved in pathogenesis. That is, if you change this region, it alters the ability of the viroid to cause disease in plants. At the bottom is an example of a genome of an avsun viroidae. This is a more extensively base paired structure, as you can see here, and more importantly, this contains a ribozyme. A ribozyme is an RNA enzyme, so basically these sequences can cleave themselves. Now, in general, viroids replicate. They're copied by host RNA polymerase II, and they replicate through production of concatemers, that is, multi-genome molecules. We'll look at a picture of that in a moment. And as I said, some of the viroids encode a ribozyme. Now, this was discovered in 1981. The fact that an RNA could actually catalyze cleavage, a cleavage reaction, in the absence of protein. So this RNA, if you produce it, can cleave itself, and it can also cleave other RNAs. No protein is involved. So this was really stunning that an enzyme didn't have to be a protein. A Nobel Prize was awarded for this work. And we like to think of ribozymes as relics of the RNA world. Remember, I've told you a couple of times that we think the first life on Earth was actually replicating RNA molecules in the organic soup that was present. And one of the molecules that evolved was probably the ribozyme. It was able to cleave itself, so these RNAs could replicate and cleave themselves via this ribozyme. No proteins were needed. They didn't arise till very later. One group of viroids, that second class of viroids that has ribozymes, they have a particular ribozyme called a hammerhead ribozyme, which is named after the way it's formed, its secondary structure. And again, this is an autocatalytic self-cleaving RNA. And as you will see, these viroids replicate by the formation of concatemers. You take the viroid genome and you make multi-mer unit molecules with 1, 2, 3, 4, 5 genomes. And the ribozyme will cleave those to form unit length molecules. So some viroids have these ribozymes built into them. Not everyone does, and the ones that don't use host nuclear enzymes to cleave the concatemers of the genome. Here's an example of the replication strategy of that group POS-P viroidae. These are the viroids that replicate in the cell nucleus. So this is a plant cell, a single plant cell, and then there's the second one next to it. And these RNAs get into the cell. We'll talk about how that happens, because this is not like a mammalian cell with a nice flexible lipid membrane. These are plant cells. They're really hard to get into. These viroids, again, they're naked RNAs. They're just moving around in the environment as naked RNAs. They get into the cell, they go in the nucleus, and there they are copied by Pol2, the enzyme that makes mRNA. And the red means it's a complementary sequence. And you make multimers of this sequence. Here we're showing just 2. That's imported into the nucleus and then the nucleolus, where there's an enzyme that will cut these multimers. So now we have 2 genome length molecules formed. They circularize, and then they're exported out of the nucleus into the cytoplasm. And then they move from cell to cell through these junctions that join up plant cells called plasmodesmata. And so you can start with an individual cell infected with a viroid, but eventually it will spread to all the cells in the plant, and the entire plant will be infected. So that's an example of what I've meant by concatemer formation. The genome is replicated to make multimers by host Pol2, and then those are cleaved by a cellular enzyme. In this case, there are no ribozymes in these viroids. So that's the POSP viroidae. The second family, the avsun viroidae, these viroids replicate in the chloroplast of the cell. That's shown in the middle of this diagram of the cell. Again, these viroids have a different secondary structure. They enter the cell, they're imported into the chloroplast, and then, interestingly, it's a DNA polymerase of the chloroplast. So chloroplasts have their own enzymes. One of them is a DNA polymerase that copies this viroid and makes an RNA copy. So you go figure what's going on there. Apparently, the DNA polymerase can recognize this viroid and make RNA copies of it. It makes concatemers as before. Now, these are cleaved, and again, these have a ribozyme in them, so they can cleave themselves into unit-length molecules. They can be replicated again to make the right polarity, because remember, the red is the complement of the genome, and the green is the genome. Now, it's just weird that you need a certain complementarity, even though you don't encode protein. But obviously, the function of this RNA is encoded in the genome strand. These get exported out of the chloroplast, and then they can go from cell to cell by moving through, again, the plasmodesmata. So this is a relatively simple replication series. They come in the cell, copies are made, and then they move on. There's no proteins involved in their genome and so forth. There's no capsid, so it's much simpler. Now, where do they come from? We discovered them in 1967, but I suspect that viroids have been around for ages. They're probably descendants of the RNA world. We know they originated in the 20th century as we were breeding crops. And when you breed crops, you start with wild plants that are in nature. And we think they were introduced into our crops by breeding with wild crops that were infected with viroids. Now, most of the plants in the world that we grow up for food, the wheat and the corn, potatoes, it's all of that. These are produced from genetically identical plant breeding lines. They're called monoculture. And so companies sell seeds or seedlings, and it's the same thing that gets grown everywhere. And so if they have the same susceptibility to viroids. And so they can all be infected, and sometimes they get contaminated at the source, and this gets spread to the farm. Now, nowadays, we try and keep our seed stocks free of viroids. We're very careful to get rid of them, but sometimes a viroid will get introduced into them. Often contaminated farm machinery can introduce them. So a tractor moving through an infected field can get viroid RNA on its wheels or on its fender and move it into another field. You can get other equipment that's used for digging or irrigation, transfer the viroids. Farmers' hands can have viroid contamination. They can move it from crop to crop, and also they can move within a row of plants from plant to plant. So they're probably been around a long time, more recently introduced, and when they come into a crop, they can cause real problems because it lowers the yield. You really can't use the crop, and you have to decontaminate your farm before you can plant again. Now, an interesting question, of course, is how they cause disease. How does a naked RNA that doesn't encode any protein cause disease? And this puzzled people for a long time until small interfering RNAs were discovered not too long ago. And these, remember, we mentioned that siRNAs are defensive molecules, intrinsic defenses in plants. They were first discovered in plants and other simple organisms. And it turns out that the machinery for generating small interfering RNAs acts on the viroid RNA. So the plant has a defense mechanism that consists of incoming pathogens being chopped up into 21 nucleotide RNAs. Those 21 nucleotide RNAs then target more incoming viruses, say, for destruction. It turns out with the viroids come into the plant and they are chopped up into 21 nucleotide siRNAs, and those then target the genome of the plant, the mRNAs of the plant, and silence specific genes of the plant, and that's what causes disease. All right, so it's funny that the very defense system, this intrinsic small RNA defense system, which is meant to prevent viruses from infecting plants, is used by the viroid to cause disease. It's not actually clear that disease is so important for transmission of viroids. It does happen, and it may be simply an unfortunate consequence. There's lots of evidence that small interfering RNAs are involved in viroid disease production. You can mutate them and show that if you remove these sequences that tend to be in the parts of the viroid that I showed earlier called the pathogenesis region, if you mutate those, you reduce the pathogenicity of the viroid, and the symptoms correlate with the production of these small RNAs. And as I said, that little region on the left in this particular viroid marked pathogenesis, these siRNAs map to that region. So the whole viroid isn't chopped up into siRNAs, it's a specific part. And as I've said, the siRNAs, 21, 22 nucleotide RNAs, derived from this pathogenesis region, silence plant genes, and that's what causes the plant disease. Okay, so that's viroids. They're, as I said, economically important. Farmers try to avoid them. We don't have any good ways of getting rid of them. We don't have viroid antivirals and so forth. So it's up to the farmers to keep their stocks, their breeding stocks of their plants clean, and if they have an outbreak, to try and contain it. But if you go online and search, you will find lots of examples of viroid outbreaks over the years. The next interesting group of infectious agents I want to talk about are satellites. And these can be single-stranded RNA, they can be DNA, they can be circular RNA genomes. But in all cases, the defining feature is that they depend on another virus to replicate. They're missing something. And the other virus is called a helper for obvious reasons, because it helps the satellite to replicate. And basically these satellite viruses, or satellites, lack genes that are needed for replication. And there are two classes that we have discovered. The first is called satellite viruses, all right? So these are real viruses. They form virus particles. They encode, the genomes encode structural proteins that actually encapsulate the genome. But they still need a helper virus to replicate because they're missing genes that are needed to propagate the genome and to do other things as well. Satellite viruses make particles that are very distinct from the helper virus. So it's in a mixed infection. And of course, these satellite viruses will not replicate unless there's a helper virus present in the same cell. So they both have to infect at the same time, and they're always transmitted together. If the satellite gets separated, it's not going to replicate at all from the helper virus. So those are satellite viruses. They actually encode a capsid. Then we have satellite RNAs. So these are kind of viroid-like in a way. They're replicating RNAs, but they're packaged by helper virus proteins, and they also depend on the helper virus for replication proteins. So these are even more defective than the satellite viruses because they lack capsid proteins. These satellite RNAs, some of them encode proteins, for example, a protein that helps the satellite move around from cell to cell in the plant. We call them movement proteins. But some of them don't encode any protein. Those are the viroid-like satellite RNAs that replicate very much like the viroids I've just told you. But again, they're packaged by helper viruses. So I've put a little picture here to show you this. This is the helper virus. The capsid, of course, is encoded by the helper virus genome. And the helper genome is shown here in green. So the green RNA provides all the functions that the satellite RNA needs to replicate, including the capsid and replication protein. So here you see the satellite RNA is packaged within the helper virus capsid. So two distinct kinds of satellites. One set They encode their own capsid proteins. We call those satellite viruses. And the other are just satellite RNAs, which are RNAs that need to be packaged by helper virus capsid proteins. Now here's an example of a satellite virus that we've talked about before, adeno-associated virus. This is a virus with a single-stranded DNA genome, which you may remember from many, many weeks ago here. It's a single-stranded DNA with the unusual T structures at either end. And the name of this virus, adenovirus-associated virus, which I've never explained to you before, means that it needs adenovirus to infect the same cell in order for it to replicate. So AAV is a satellite of adenovirus. Adeno is the helper virus. It provides a number of functions to AAV. Now, AAV encodes its own capsid proteins. That's why this is a satellite virus. So the AAV capsid shown at the upper left is different from the helper adenovirus. But again, for AAV to replicate, it has to be co-infected in the same cell with adenovirus. Now, what does adenovirus provide? This is very interesting. What AAV needs are the early proteins of adenovirus and the E1A and other proteins of the early region 1 area of the adenovirus genome. And there are at least two reasons why AAV needs these. First, some of these adenoproteins activate the AAV promoters so that they can function. But does anyone remember another function of the E1 region of adenovirus that's important for adeno replication? I'll give you a hint. Its target is RB. Remember that? You should. So you're going to have to know it soon. Okay. E1A inactivates RB and P53. And what does that do to the cell when you inactivate RB and P53? It goes into mitosis because adenovirus wants the DNA replication system of the cell to be activated, right? That's why AAV needs the E1 proteins as well. AAV has no way to kick the cell into mitosis. And so it does that via the E1A proteins, so it can replicate its genome. And then in the end, you have distinct particles made with AAV. These are AAV capsids and just the AAV genome inside. And the adeno is packaged separately. So that's an example of a satellite virus. And the cool thing, and we'll see in next week, the last lecture, we're going to talk about gene therapy. AAV is a huge virus for doing gene therapy because you can make stocks of AAV free, and you can infect cells without a helper, so they deliver the gene, but they don't replicate. And that's really good for gene therapy. And many people are using these. We'll talk about that in a week. Now, satellites in plants cause distinct disease symptoms that you don't see with the helper virus alone. So AAV is obviously not a plant satellite. So these kinds of molecules go beyond plants. Satellites can infect plants and mammals. But when you have the satellite and the helper, you get sometimes a different disease. So helper virus in plants may cause a certain disease, but it's different in the presence of the satellite. So here is an example of a yellowing. This is called chlorosis. When the plants lose their chlorophyll, they get these yellow areas. And this is caused by a specific satellite together with a helper virus. Remember, you have to have both because the satellite can't replicate without the helper. Necrosis, of course, is destruction of the leaf. Now, again, satellites have no homology with the helper virus. Whether it's an animal satellite like AAV and adenovirus, AAV genome has no homology with the adenovirus genome. And the same with the plant satellites and the plant satellite helpers. They have no homology, and the satellites are not simply defective viruses derived from the helper. They're distinct entities that have evolved, but always in a cell infected with a helper virus. Quite an interesting situation, but in biology, these things can arise if opportunities are presented. I just wanted to show you 3 different satellite viruses. Again, these are not satellite RNAs. These are viruses, satellite viruses that encode their own capsid proteins. So for example, we have adeno-associated virus. The helper, I've told you, is adeno. It can also be herpes virus, can provide the same functions in a helper situation. And that's the genome of AAV. It's single-stranded DNA, the particle size, the length of the genome, and the capsid proteins. These infect vertebrates. Then there's another one called chronic B paralysis virus satellite. So the way these are named, there's the virus, which is a helper, chronic B paralysis virus, and then the satellite is CBPV satellite. This is a single-stranded RNA satellite virus with a smaller particle, small genome, and just one capsid protein. Infect bees, obviously, and one of them that infects plants, and in particular tobacco necrosis virus, and tobacco necrosis virus satellite. And you can see that's also a small capsid as well. So again, the point here is that tobacco necrosis virus, by the name, it can replicate on its own. It doesn't always have to have a satellite with it. It causes a certain disease of tobacco. But when the satellite is present, you get a different disease. It can be more virulent. It can be just different in its features and so forth. So the satellite modulates the disease. And these are both problems for agriculture. So there's one animal satellite virus I want to tell you about. It's hepatitis delta virus. And we have hepatitis A, B, C. They're named in the order that they were discovered. And there's a hepatitis delta, there's a hepatitis E, and then we skipped F. I don't know why, we went right to G. You can guess why they probably skipped F, the effing virus, right? This delta, hepatitis delta, has properties both a viroid and a satellite. I'll show you why. Its helper virus is, interestingly, hepatitis B virus. And we think that, you have Hep B, you get a certain liver disease, you may progress to hepatocellular carcinoma. We think if you also get delta along with your Hep B, remember, delta is a satellite. It cannot replicate without Hep B. So you can never be infected by delta alone. You always have to get both. We think the disease caused by both B and delta is more severe than with hepatitis B virus alone. I mean, we don't really understand that, but that's the observation. Here's the global distribution of hepatitis delta virus. You can see here essentially Asia and parts of the Middle East, and the highest prevalence are the dark. So Japan is has a lot of Hep delta, for example. 18 million people are estimated to be infected with HDV. Now, of course, they also have Hep B. They can't have delta alone. But it's about 5% of the 350 million people who have hepatitis B virus. So they also have delta, and they tend to have more severe disease. It used to be higher in Europe. It is declining in Europe, very prevalent in the Asia-Pacific region, which goes along with the prevalence of hepatitis B virus, of course, and it needs Hep B in order to replicate. This is the genome of delta. So it's viroid-like in its genome, in its shape, and the fact that it's rod-like, single-stranded RNA. It's satellite-like in that it encodes proteins and needs a helper virus. So that's why I say it combines viroids and satellites. So at the top is the genome. It's a 1.7 kilobase single-stranded RNA, but mostly base-paired. So it looks like a rod in the EM. And typically in a liver cell, there are about 300,000 copies of this delta genome. And then there's what we call an anti-genome, which is simply the complement of the delta genome. And the important point about this is that there's an open reading frame. It's shown in orange here in the delta genome. And there's a self-cleavage site. So this RNA also has a ribozyme in it, like some of those viroids do. From the genome, you make a single mRNA. So the anti-genome would be the plus sense, if you will. It's the same polarity as the mRNA. You make a single mRNA that encodes for a protein called delta antigen, and there is actually an editing site at the end of the genome so that you can make two different sizes of delta. You may remember RNA editing is when you add extra bases which are not encoded in the genome. It's caused by stuttering of the polymerase. So that happens here at this editing site, so you get two different proteins that are made. And that's the only protein made in delta-infected cells. It's the only delta-specific protein. And again, it's a ribosome, and the ribosome sequence is shown in red here. Again, on the top is the delta genome, and of course this is packaged by capsid proteins of hepatitis B virus. On the right is hepatitis B virus. Remember that crazy partially double-stranded DNA genome with a protein and a small RNA linked to it. as we learned about a long time ago. It's packaged in a capsid, which in turn has an envelope around it, and then the envelope is embedded with glycoproteins. And when you make a hepatitis B vaccine, you just produce these glycoproteins, and it makes a virus-like particle vaccine. The delta virus particle is basically the viral RNA that's shown here in the middle, so that base pair genome is in here, and it's encapsulated in a membrane comprised of the hepatitis B virus proteins. You can see large, medium, and small hepatitis B proteins. They encapsulate the delta, and then the small and large delta antigens are thought to help package the RNA into this particle. So you can see the large delta antigen is stuck in the membrane, and we think that attaches to the RNA. And then the small delta antigen almost acts like a nucleoprotein and coats the entire RNA. So those two delta antigens are essential for getting the delta RNA packaged into the membrane with the hepatitis B surface antigens. So that's the helper function provided by Hep B. Now, when this RNA enters a cell, here on the upper left is the hepatitis delta RNA genome. It's replicated by host polymerase II. And this is the only RNA agent that we know of that's replicated by Pol II. So this is an enzyme that's normally recognizing DNA in our cells and making mRNA. It actually can copy the delta genome. So this is the only exception to the statement I made way back at the beginning that we have no enzymes in our cells that can copy RNA virus genomes. Because this is not a virus, this is a satellite. But nevertheless, this circular RNA is copied by DNA-dependent RNA polymerase. Even though it's an RNA template, it apparently can be recognized by Pol II. When that RNA comes into cells, there's the minus strand RNA genome. Then Pol II recognizes it and begins to make a complement. That's shown in green. And you make concatemers, just like for the viroids. You make multiple unit length molecules. Now, the red is the ribozyme cleavage site. And so that generates unit length or genome length molecules. So here we're showing one of these molecules being cleaved by the self-cleaving ribosome in red. And that gives rise to mRNA and gives rise to the delta antigen. But just processing at the ribosome site only without the production of an mRNA gives rise to individual genomes. So here in 9 and 10, we have full-length plus-stranded RNAs, which are the complements. Those are then copied again by Pol II to make the opposite strand, shown in the olive color. And again, those have ribosome sequences in them, so they can self-cleave to generate unit-length genomes, which then circularize. So that's the replication strategy. Again, carried out entirely by host Pol II, which was proven just about 5 years ago or so. People for years were puzzled about what was copying this, and then they found that it was Pol II that could copy this RNA, which was certainly not expected. Now, one more nucleic acid-based unusual infectious agent, and these are what are called virophages. I don't really like this name. It's derived from bacteriophage. Okay, phagein in Greek means to eat. So this really means virus eater, viro eater, virus eater, which is a stupid name, but I didn't give it to it. These are circular, double-stranded DNA viruses with icosahedral capsids. And very much like the satellites that we've just talked about, they only replicate in cells infected with a helper. But in these cases, the helper is always a giant DNA virus, like Mimi viruses and Pandora viruses that we've talked about in this course. And what they do is they interfere with the replication of the helper. So when these were first discovered, they were found to knock down the yield of the helper virus, and that's why they were called virophages, to give this idea that they're interfering with them, but they're not actually eating them, right? They're just interfering with their yield. That's why I don't think it's a very good name. But unfortunately, the first person to discover something, they get to name it, and usually it sticks. And in this case, the first people to discover these were French, and they're very persistent, and so the name is stuck even more, virophages. Here's a table of just some virophages that have been identified. On the bottom, you can see a map with little dots showing where some of these different virophages come from. And you can see they're all over the world, not only on land but in water. And you may be wondering, how do you isolate a virophage all the way in the middle of the Pacific or down by Antarctica? Well, you don't. You actually just look at the global ocean data set. This is basically, many years ago, Craig Venter, the guy who first sequenced the human genome and who is incredibly wealthy and had started his own institute. Guess what the name is? The Craig Venter Institute. He took his boat, his yacht, and he sailed around the world, and he sampled seawater and sequenced it. And you can access those data. They're all online for you. And after they identified the first virophages, people went back and mined that data, and they found tons more of virophages in all of the data sets. That's what most of these dots are. But some people have actually isolated some of these viruses. The first one was Sputnik, which was isolated as a satellite, basically, that requires a mimivirus as a helper virus. And this was from a cooling tower in Paris. Ma virus was isolated in the coastal waters of Texas. Its host, its helper, is a virus called Cafeteria roenbergensis virus. And the virus infects a flagellate in the ocean, a marine phagotropic flagellate. And then organic lake virophage, which is a lake down in Antarctica, a hypersaline lake. And then another one, which is pretty interesting, it's called Sputnik 2, was isolated from the contact lens fluid of a patient with keratitis, inflammation of the eye in France. And it is, its helper was called lentille virus. You know what lentille means? Lens. It means lens in French because it was isolated from a contact lens. So they do have a sense of humor over there. And so these are the eukaryotes that the virus, helper virus, infects, and then the virophage depends on the helper for replication. You can see also from Yellowstone Lake and another Antarctic lake. Now, these are pretty big. Look at the size of the genomes, you know, about 20, 30,000 base pairs with quite a few proteins. These are way bigger than the satellites of plants and hepatitis delta. So they're something in their own. And that alone makes them quite different, simply their size. And here's a photograph on the right of the structure of the first virophage Sputnik that was discovered. You can see it's an icosahedral capsid with many subunits. So this could be a bona fide virus, except that it depends on a helper for replication. Now, you know, up until now, people have called these satellites, but the people who work on them are having a big fight with the rest of the virology community, because they say, you know, you call adeno-associated virus a virus, and it's smaller than some of these virophages. We want to call these viruses too. Here's what the Sputnik virophage does to a mimivirus infected cell. So these big particles are mimivirus. So on the upper left is a mimivirus infected cell. These big icosahedral particles are mimiviruses. And here is a mimivirus factory. These viruses, again, these are giant viruses of amoeba. They are assembled in factories in the cytoplasm. And the smaller particles you see on the upper left, those are the Sputnik virophages. They replicate within the factories where the mimivirus are replicating and being assembled. Makes sense, they have to be where the mimivirus is, because mimivirus isn't elsewhere in the cell. So these little mimivirus, these Sputniks are replicating there. And in fact, what they do is they screw up. As I said, they lower the production of infectious mimivirus, but morphologically they mess up the particles. So for example, on the upper right, you can see a mimivirus, and it's got virophages inside of it. So these virophages get inside the mimi particle, and that's obviously not good for infection. They may actually be replacing the DNA. Panel C is another example of that. And panel D, so normally these mimiviruses have multiple concentric layers of capsid protein on top of the genome. And look at this one, it's all screwed up. The layers are on one side of the particle rather than all the way around it. Here's another example at the bottom. And on the left, you can see this. These layers actually peeling off the particle. See, these are all from cells that are co-infected with the Sputnik virophage. So they reflect the effect of the virophage on the morphology of the particle. And this is presumably why there's less infectious mimivirus in cells infected with these Sputnik virophages. So not only do they need the mimivirus to replicate, they also inhibit their production. Now, what are these doing in the environment? There's some very interesting clues about the function and the role of virophages. One of them that's quite interesting is called Ma virus. It is a virophage of a virus. It's a giant virus that infects Cafeteria rowenbergensis. This is up here in the upper right, Cafeteria rowenbergensis. It's a flagellate that lives in the oceans, and it eats things. And it eats a lot of different things. That's why they call it cafeteria. You get it? Because it comes in with a tray and goes out with everything and eats it all. And this Ma virus is a virophage that depends on this Cafeteria rowenbergensis virus. The other one I mentioned already is organic lake virophage. This organic lake is in Antarctica. And this virophage depends on helper viruses called phycodnaviruses that infect algae. Now, one of the clear roles of these virophages is as gene exchangers. You can find sequences in their genome that match exactly sequences in the helper virus genome. So we think these virophages grab by recombination genes from their helper and move them to other hosts. And we have evidence that they have moved genes from cell to cell that way. They also seem to have an impact on ecology of not just the ocean, but also of some of these lakes in Antarctica. So they actually protect populations that are infected by the helper virus. So in this lake in Antarctica, organic lake, these are freshwater lakes. They have a very, very simple population structure. Not a lot of microorganisms because it's dark most of the year and there's not a lot of nutrients. And these virophages seem to protect the population of algae and other microorganisms in these lakes that are otherwise going to be destroyed by the helper virus. So these virophages probably have a big role that's underappreciated in global ecology. As I mentioned before, some people who study these are offended that they're not considered a virus because they're big and they have a big capsid and have 20 or 30 genes. And they say, like many autonomous viruses, these virophages depend on the transcriptional machinery. The only difference is that in their case it's from another virus, whereas for other viruses, most of the viruses we talked about, these DNA viruses, depend on the transcriptional machinery of the host cell. We don't call those satellites, right? We call them viruses. They're parasites of the host cell. And they're saying here, these virophages depend on a virus-infected cell for the transcriptional machinery. So they think that they should be afforded actual virus taxonomy rather than calling them a satellite. They're really complicated, and they just need the transcriptional machinery of another virus. So basically what's happening here is that the classification of these virophages is changing. But I think it's a fundamental issue. And this is a good argument that these virophages, just like most other viruses we talk about that are not defective, they need transcriptional machinery. The source, that shouldn't make any difference in calling it a satellite or a regular virus. So lastly, I want to talk about prions. And here is where you learn not to eat meat anymore, okay? Or at least beef, because we've learned not to do a lot of other things here, and now we are going to get to beef. And I just leave this to the end. Prions, which are proteins that are infectious. They have no nucleic acid. This is the amazing, remarkable story. And you may be thinking, why are you talking about this in a virology course? And the answer is, in the beginning, the people who studied these prions, no one would listen to them except virologists, because they moved around in populations like viruses did. They were infectious. And so now they're in the virology textbooks. They've kind of ended up there, even though they're just infectious proteins. And they're really, really cool anyway, and you want to hear about them. And you've heard about them in the news: bovine spongiform encephalopathy, mad cow disease, Creutzfeldt-Jakob, Scrapie, Kuru, chronic wasting disease. All these words you'll see in the press. And in fact, in 1997, Stanley Prusiner got the Nobel Prize for discovering prions. And I found this picture on the internet. It's actually a road, I think it's in the UK, maybe not. It's called Prion Road. Boy, I would like to live there. That would be a great address, right? 100 Prion Road. Prions cause diseases that are a little bit long named: transmissible, spongiform encephalopathies. All right, let's break that up. Encephalopathy is just a disease of the brain. And remember, encephalitis is an inflammation of the brain, so that's different. So an encephalitis is a kind of encephalopathy. These are fatal, always fatal, neurodegenerative disorders of mammals. not just humans, but other mammals as well. Thousands of humans each year, and about 1% of these arise by infection. We'll talk about how the others come up. So for example, by 2002, 120 people had contracted Creutzfeldt-Jakob disease from eating meat from animals with mad cow disease. And the thing is, you can't tell that the cows have mad cow when you eat them, and it's not till many years later when you start getting symptoms that you know you have the disease, and by then it's too late to do anything. TSEs occur not only in humans but in animals. So here are some animal TSEs. The one we'll talk about in a bit is bovine spongiform encephalopathy, or BSE, or mad cow disease. And we also have another one that's been in the news a lot, chronic wasting disease. This happens in deer, elk, and moose. Those are cervids, I believe. Exotic ungulate encephalopathy, Nyala and greater kudu. I think this is a Nyala. I didn't know what a Nyala was, but we don't have them here. But they can get these TSEs. Of course, we do have deer and elk and moose here in the US. Feline spongiform encephalopathy. Your pet cat could get it if you fed it contaminated food. And great cats out there, we've given it to great cats in zoos by feeding them contaminated food. Scrapie in sheep and goats, and then minks get it as well. And there is actually a reason why all these animals get it, which we'll touch upon later. The human TSEs include Creutzfeldt-Jakob disease, fatal familial insomnia, Gerstmann-Straussler syndrome, Kuru, and variant CJD disease. I remember watching some TV detective show once, and the guy said, "It's fatal familial insomnia." First time I've ever heard that, which is a prion disease on a TV show. Okay, transmissible spongiform encephalopathies. Spongiform refers to the fact that the infected brain looks like a sponge. It has holes throughout it caused by infection. This disease is accompanied by a number of different conditions, and sometimes the actual strain of prion makes a difference, but they include psychomotor dysfunction, and each disease has a characteristic symptomatology. You can have depression, you can have getting very angry, really depends on the part of the brain that's affected. Here's a slide of a brain section showing you the spongiform appearance. See all these white areas? They're not supposed to be there. It's supposed to be all brain matter, neuropil. But here the TSE has resulted in the destruction. There are also these dark staining tangles. Now this all began with sheep. Scrapie was a disease in sheep that was the first TSE recognized. And this disease was called Scrapie because the sheep rubbed themselves on fences, and their fur or hair, I don't know what sheep has. Does anyone know? Fur? Wool, thank you. Wool. Yeah. Every year I forget. Wool. Their wool comes off and leaves the skin showing, so they call this Scrapie. And this is accompanied by motor disturbances. They have trouble walking. You see a Scrapie sheep, they're wobbling back and forth. Trembling. They have uncontrollable trembling in spurts. In French, in Canada, I believe, is called tremblement du mouton. Paralysis, weight loss, and they die within 4 to 6 weeks. And that was known for 250 years in Europe. And in the UK, you still get 1% of sheep a year coming down spontaneously with Scrapie. So if you go to the UK, you should not eat mutton, right? So the sheep farmers found, these were the first epidemiologists, they found that they could transmit the disease to a healthy herd. They would take their sheep and give it to their friend down the road, and the sheep would transmit the disease. So it was clear that it was infectious. So people started studying what was infectious in the sheep, and they found by 1939 that you could grind up sheep brain and filter it through a filter that would pass only viruses, a 0.2 micron filter, remember, from the very beginnings of this course. So they said, yeah, it must be a virus that's going through a filter. Grind up a sheep brain, filter it, take the filtrate, inject it into another sheep. The sheep gets disease. Could be a virus. But it turned out to be way more resistant to UV, ionizing radiation, formaldehyde than for any known virus. So it was being questioned whether it was in fact a virus. And eventually, the high resistance to all these agents led to the idea that there was no nucleic acid in it. Now, this was very controversial, as you might guess. And up until just a few years ago, people were still skeptical. They said, there's a little bit of nucleic acid in that protein. But now we're quite clear that there's no nucleic acid in Scrapie. And you know, when the first experiment was done in 1944 by Oswald Avery McCarty, showing that DNA was the genetic material, people said, you have protein contaminating your DNA. So in science, there are always skeptics like this, and you have to just get around that. Here's the experiment showing the high resistance to radiation. So we're looking at ionizing radiation here in rads, and this is the amount of dose from 10 to the 4 to 10 to the 7th in rads. And then here we have the molecular weight of the nucleic acid, and these are inactivation curves essentially. So on the top we have a bunch of DNA viruses. We have big ones, herpes simplex, and big vaccinia up here. And then as we go down, it doesn't take a lot of radiation to knock out the infectivity of these genomes because it's a big target. So you need less dose to knock out the infectivity of herpes and so forth. So this is the amount of radiation you would need to inactivate infectivity of herpes. And that's the molecular weight of the genome, and this is the size of the genome in kilobases. And then as the genome gets smaller on this top line, you can see SV40 and polyoma are much smaller than herpes genomes. It's harder to knock them out. You need more radiation. You need almost 10 to the 7th rads, and that's because the target is smaller. And the radiation is not likely to hit it when the genome is so small. And on the bottom line is RNA viruses. You have large RNA viruses on the left here, like yellow fever, about 12 kb or so, all the way down to smaller RNAs down here. Here's tobacco necrosis virus, which you were just talking about in terms of a satellite. And again, the inactivation, you need more rads as the genome gets smaller. But here's Scrapie. It was more resistant than any DNA or RNA virus that had been determined. And you can see that it's down here with both the DNA and RNA viruses. And again, this was one of the pieces of evidence that suggested to people there was no nucleic acid in the agent of Scrapie, whatever it was. Going back to TSEs, it was, as we studied TSEs in sheep, we developed animal models, and we found that the animal and human TSEs, so we recognized TSEs at this point in people. There were several shared abnormalities, defects in plasma membrane formation, vacuolization, as I showed you on that one slide, loss of neurons, the spongiform appearance, the accumulation of glial fibrillary protein in clumps, and amyloidosis, that is, fibrils of amyloid precursor protein. So in animal models of Scrapie and human TSEs, they had lots of similarities. So people were thinking that they were the same disease. We could reproduce TSEs by injecting organ homogenates into animals like hamsters and mice. This would result in difficulty walking, dementia, and death after many years. And when you inject the virus, it accumulates in the lymph cells, in the lymph organs, and then spreads to the CNS. And then in the CNS, the protein accumulates and causes the typical spongiform appearance of the disease. Now, very interestingly, this is an infectious process, but there's no inflammatory antibody or cellular response. No immune response whatsoever to in these TSEs. And that should tell you probably what's going on. Now, these TSEs, up until recently, and in fact in humans, still very hard to detect. You usually detect them when you get symptoms. People start having psychomotor issues, difficulty walking, cognitive issues, many different things, and then they can be diagnosed. Now we have a molecular test that we can use to diagnose them. But we don't screen populations for this disease at all. They're untreatable. There's no way to alleviate any of these symptoms, and they're invariably fatal. So these are very serious diseases, although fortunately they're rare. Back to the Prion story, remember we had Scrapie being transmitted to sheep, high resistance to ionizing radiation. A mathematician suggested in 1967 that the agents were pure protein, and then Prusiner purified the protein. He used biochemical processes to purify it and showed that you could transmit it to animals. Now, he used old school purification where you would run columns and fractionate Scrapie brain extracts and highly purify the protein as much as you could, and then inject it into animals and show that it causes Scrapie. But again, the skeptics said you have nucleic acid contamination in there, and it wasn't until we could clone the gene for the protein that we could exclude that possibility. So he's the first to call the agent a prion, a proteinaceous and infectious particle. And he also cloned the gene encoding the prion protein. It's called PRNP, prion protein gene. And this is essential for the pathogenesis of TSEs. We now know that in mice, for example, if you delete the PRNP gene and you inject Scrapie prions into mice, they will not develop disease. So the gene within us is actually what is causing this disease. So our current view is that the Scrapie in sheep, or the protein, we have now called the pathogenic protein PRPSC in honor of the sheep. That pathogenic protein is a conformational variant of a normal protein that we all have called PRPC for cellular. And this protein is found on the outer surface of neurons. It's GPI anchored. And if you take the abnormal conformer, like the Scrapie prion, and you inject it into the organism, it converts PRPC into the pathogenic conformation. So we all have PRPC in us, mostly in our CNS. If I eat some PRPSC in the form of an infected cow or infected sheep, that PRPSC will get into my CNS, and it will cause my PRPC to turn into PRPSC, which is a pathogenic form. It precipitates and causes all the CNS symptoms. Here that is summarized in the diagram. In the middle is, here at the top is PRPC, the normal cellular prion protein. And you can digest this with an enzyme called proteinase K, and it chops it all up into bits. This is an early diagnostic test. PRPSC, this is the pathogenic form of the protein. Same amino acid sequence, but simply is folded differently. You cut that with proteinase K, it only nips a little bit off the N-terminus. So early on, people did Western blots after protease digestion to show if an animal had PRPSC in them. And on the right is a structural view. On the top, the normal PRPC protein and the PRPSC conformational altered protein. You can see lots of beta sheets, and that's part of the difference between the two proteins. So again, it's the same protein in sequence, just that PRPSC, which is associated with prion diseases, is simply folded differently. Again, we all have PRPC, normal PRPC, which is folded properly. If we acquire some PRPSC, it'll cause the conversion of all our PRPC into the pathogenic PRPSC. Now back to mice. If you take both copies of the PRNP gene away from a mouse, you can easily do that. They're resistant to infection. So you need to have the PRNP gene in order to develop a prion disease. You can introduce PRPSC into an animal. So you can feed mice or inject PRPSC and they will develop a TSE. Or sometimes rare mutations in the gene in people lead to spontaneous development of a prion disease. But in either case, if you inject or if you have a genetic predisposition, the pathogenic protein accumulates in the central nervous system, and that causes the TSE. There are three types of TSEs, actually spongiform encephalopathies. There's infectious or transmissible, which was the first discovered, so it was called TSEs, but they're not all infectious. There's a familial or genetic spongiform encephalopathy. We'll go into these in a bit. And then there's sporadic spongiform encephalopathy. And if an animal develops any of these, the disease can be transmitted to another animal by giving them diseased tissue. Kuru is an example of an infectious or transmissible form of the TSE. We'll talk about that in a moment. A number of people have gotten TSEs by receiving corneas, hormones, or blood from patients with Creutzfeldt-Jakob disease. So this has a very long incubation period, and you can donate material, or if you die and people don't know you have Creutzfeldt-Jakob, you can give corneas or blood or organs, etc., to other people. And we have seen transmission of a TSE by that way. We've seen people getting, we've had cows getting a TSE by feeding them ground up sheep. We'll talk about that in a minute. And people eating the cows who got sick from eating sheep have developed CJD. Those are all infectious. You eat the contaminated material and you get the infection. Now, the first human TSE study was called Kuru, which was a fatal TSE found in the 4 people of New Guinea with a 30-year incubation period. And this was very, very prominent in this area. So the US sent a virologist named Carlton Gajdusek to study this. And he found out that the reason it was spreading is because when someone died in this 4 people, the women and the children get to eat the brain of the dead person. And so women and children would get TSEs, but not the men. And so he said, you should stop eating your dead relatives. And eventually they stopped, and that got rid of this disease called Kuru. So it was spread from human to human by cannibalism. Because again, the people would presumably die of a sporadic or genetic TSE. And of course, we didn't know what they had, and then their brain was removed and eaten by the women and children, and they would spread the disease to them. So this provided a lot of information about the nature of these diseases. That's a transmissible spongiform encephalopathy. There's a sporadic form. It affects 1 to 2 per million people worldwide, typically 50 to 70 years of age. And this accounts for 65% of the TSE that we see globally, sporadic. It appears with no warning or epidemiological indications that the PRNP genes of these people are normal. And this has been transmitted to other people by donating lenses or blood and so forth. It's even been transmitted by ocular equipment. You know, an ophthalmologist looking at a patient's eye, touching metal material to the eye, gets contaminated with prions. You can't autoclave these things. They're resistant to autoclaving, and then it gets transmitted to another patient. And we think Kuru may have been established by the first sporadic case, and then they ate his brain, and that spread it through the population. The protein must be sporadically misfolding in your CNS because we don't see any genetic predisposition to this. We do have a familial spongiform encephalopathy, which is inherited, and this is associated with mutations in the PRNP gene. And again, this has been transmitted to other people through organs, corneas, blood products, etc. So these are all transmissible. So here we have a mutation in the gene encoding the prion protein, which after so many years is allowing it to misfold and become the pathogenic form. And as soon as you have a little bit of misfolded protein in your CNS, then that catalyzes the misfolding of more and more normal protein, and then the disease takes over. And you can tell because you can sequence the PRNP gene from these individuals and see what they have. Now, at some point, all our genomes will be sequenced at birth, so you'll be able to know right away if you have a mutation. And there are only so many mutations in the PRNP genes associated with this disease. You'll be able to know if you have the disease. But right now, we can't do anything about it. We have no way to prevent it, and we have no way to fix the gene. But presumably, we'll have some therapies at some point. These are the cases of Creutzfeldt-Jakob disease. This is the sporadic form, which accounts for most of the TSEs globally. You can see this is US from 1979, about 175 cases. 2011, over 400 cases. The red is the age-adjusted rate, so it's very constant within a certain age frame. But the incidence is going up, as you can see. These are the 3 kinds of TSEs again. I just want to review for you. We have all a normal PRPC. You can eat contaminated material with PRPSC. You can get it through a transplant, a cornea. You will get an infectious form where your normal protein misconverts. You can have a mutation which predisposes you to the conversion from C to SC, or you can have the sporadic form, which is spontaneous misfolding. Now, if you eat contaminated material, we think the PRPSC goes into your intestine, converts the PRPC in your intestines to the pathogenic form, and then that travels by axonal transport up into the CNS and continues its propagation there. And the other forms that you would acquire spontaneously or through genetic forms, again, the protein is misfolding somewhere. We don't know exactly where initially. It could be in the CNS. Now, there was an outbreak of BSE in the UK not too long ago, and this was a consequence of feeding cows the meat processed from sheep who had Scrapie. So it's a form of cannibalism, just like the Kuru outbreak. What they used to do was grind up dead sheep and feed them to the cows. You feed cows protein, they grow faster, as opposed to letting them go out in the pasture and eating grass. And they do this in Europe, where there's not a lot of grazing area. And in the 1970s, the method of preparing this feed, which is called meat and bone meal from the ground up animals, it changed and allowed the Scrapie proteins to pass into cows. So previously they had a step in the processing which got rid of the Scrapie protein. So there was no Scrapie in cows up until this time when we changed it, probably to save money. And the cows started developing BSE, which was a form of transmissible spongiform encephalopathy. They stopped feeding them and they stopped the outbreak. But unfortunately, those cows had already gotten into the human food chain, and there was an outbreak of a brand new TSE in humans called variant CJD of people who ate BSE beef. About 1 to 2 million cattle in the UK were infected with prions. And the problem is that the incubation time before you see the cows start to stumble and fall takes about 5 years. And unfortunately, you kill a cow at about 2 or 3 years of age for meat. So we were introducing contaminated beef into the food chain. So here's the outbreak of BSE in cows in the orange. And we changed the way, so we saw initial outbreak, we changed the process of making their feed, and we got rid of the outbreak in cows, but that was followed by an outbreak in humans of variant CJ, which is the blue bars here, and eventually we got rid of that as well. But it went from sheep to cows to humans. This is all about prions in the food supply. In fact, today, new cases of BSE in cattle occur everywhere, and they're all sporadic. So we're not feeding cows dead sheep anymore, but the cows develop their own sporadic TSEs just like people do. So we try to look for this in cows, but we only look at 2% or less of the slaughtered cows in the US. We could look and see if there are prions in them, but we don't do that. Now there's some diagnostic tests which may be used, and eventually we'll have some drugs that can block accumulation of prions, and maybe we can treat people with those, but those are a ways off. Here are the cases of BSE in North America alone. These are after the ban on preparing meat in a way that would transmit it. So these are all sporadic BSE cases. You can see the US is orange, Canada is blue, and these blue ones are all after the feed ban, so that means they're sporadic. So these are just a few cows every year, 1 or 2 or 3 or 4, but again, this is because the cows have developed symptoms of disease, so they've lived a bit longer, but if you kill them earlier, you don't pick this up. So we're very concerned about introducing BSE into the human food chain, and so we've developed a diagnostic test to pick this up. I have 2 articles here if you're interested in reading further about this. The top is a case of prion disease from contaminated beef. This is a gentleman who apparently ate beef. They traced this back. He ate beef in a country, in a Middle Eastern country that had gotten beef from the UK during the this BSE outbreak, and he came to live in the US, and at about 60, 65 years of age, he slowly developed dementia and inability to move, and he died of a BSE. But they could trace back that he had eaten this, and of course he had no idea that he had done this. The other one is prion contamination in the emergency room. This is a case in the US of a man who shot himself in the head, attempted suicide, was brought into the emergency room still alive, of course his leaking brain and blood out of the wound, and he died, but later they found he had a prion disease, and he was starting to get a little demented, and that's why he shot himself. They had to decontaminate the whole emergency room and destroy lots of equipment worth many thousands of dollars, because this is a real risk to other people. And that's really scary, because he came in, was treated, died without anyone knowing for a few days that he actually had a prion disease. This is the diagnostic test we have now. We can take a brush of the olfactory neuroepithelium, because if you have prions in your brain, you or a cow, they will get into your olfactory neuroepithelium. You can brush them and sample it. Then you do a kind of a amplification reaction, sort of like PCR with protein. You take some PRPSC, which presumably comes from scraping. So if there's some oligomers of prion in there, you mix them with fresh prion C protein, and then these will form longer and longer chains in vitro. Then you break them up by sonication, and you keep repeating this. You get more and more. You get an amplification of the misfolded prion, which then you could test on a gel. And this has turned out to be reasonably good for detecting infection in people and in cows. If you inoculate prion material, typically you need to put it in the same species to cause disease. If you put a different species into the brain, it's inefficient. So in other words, the sequences of the inoculum, the PRPSC, and the recipient have to be the same. So we have a species barrier which limits the transmission, and that's in the sequence of the protein. For example, if you inoculate hamster PRPSC into mice, you don't get disease because there's a difference between the hamster and the mouse protein. However, if you make the mouse transgenic for hamster PRNP, then inoculating PRPSC from hamsters will result in disease. So it's a nice way to test the risk of any particular species. How about cows? Unfortunately, if you take cow PRPSC and inoculate it into mice, you get disease because cow PRPSC has a wide host range, and that's why cow PRPSC was able to infect humans. And that's why we're afraid of BSC still in cows. Now, a new disease has arisen since 1965 in deer and elk within North America and also Korea. This is called chronic wasting disease, which is a TSE of deer, elk, and moose. And you can see the Scrapie-like appearance. Standing herds up to 90% are positive, and wild cervids up to 15%. So there are lots of deer out there that are infected with TSEs. Same thing, a prion disease. These deer shed the TSEs. In nasal secretions, saliva, it's on their skin, blood, milk, birthing matter, dead carcasses, and they can all infect another animal. For example, this deer is licking the grass that's been contaminated with another deer's TSEs. It can be ingested, it will replicate in the cow. And this is how these TSEs are spreading throughout deer in the northern US. It recently was found that grass plants can bind, retain, uptake, and transport infectious prions. So they did an experiment where they contaminated the soil with prions in red, and the prions are taken up, transported up to the leaves, and then they could feed the leaves to animals and get prion diseases transported. They can also show that prions stick to the leaves and can be transmitted to animals as well. So in other words, a deer which is excreting prions, it's going to contaminate grasses, and they could be transmitted maybe days later to another animal. So is chronic wasting disease have a broad host range? If you put a cervid PRPSC into mouse, no disease. But if you make the mouse transgenic for cervid PRNP, you infect with PRPSC, you do get disease. So it looks like this has a narrow host range. However, if you take transgenic mice that have the human PRNP gene, you infect them with PRPSC, no disease. So this sounds like it's good that cervid PRPSC should not be able to infect people. But of course, this is a mouse, and mice are not people, so you always have to worry about what could actually happen. Unfortunately, take deer PRPSC, you infect cow, you get disease. So we're worried that deer will contaminate pastures. If cows are grazing in them, they'll pick up prions from the deer. The deer prions will get in the food supply, and then we will eat the cows and get this chronic wasting disease indirectly. So we're very aware of this. If you're a hunter, you can go to cwdinfo.org, and they teach you how to dress out a deer if you shoot it in the field. You know, stay away from the spinal cord, use gloves, don't eat the brain or spinal cord tissues. So far, there has not been a case of acquired TSE from a hunter killing a deer, but it's only going to be a matter of time if this TSE can in fact cross from deer into people. I think all of these TSEs are related, and they probably began a long time ago with the development of Scrapie in sheep. So on this diagram, we're looking at naturally occurring disease in blue. So I think sheep develop Scrapie naturally, and it probably has spread to a number of other animals. It's probably spread to deer and elk. Maybe that's the origin of TSEs in deer and elk from sheep, or maybe it was a spontaneous disease in them as well. I think minks have acquired the disease by feeding on animals that are infected, like sheep. We fed sheep to cows, and we caused an outbreak of BSE in cows, which was then transmitted to other cattle and transmitted to lions and tigers in zoos. We feed them cow parts, and those were contaminated with BSE. We fed them to primates and perhaps ungulates in the wild as well. And some cat food has been contaminated with BSE beef as well, and that spread TSEs to cats. And of course, we ate contaminated beef. We started the variant Creutzfeldt outbreak, and that has been transmitted to other people in an iatrogenic form, either in hospitals or by transplants and so forth. And where did the human disease occur? We think perhaps there are some sporadic cases that occurred a long time ago. and those have led to contamination of donated materials and perpetuation of Creutzfeldt-Jakob as well. It's a very interesting disease biologically, but a little bit scary. A few students have in the past told me this is why I'm a vegetarian.